Yes, you've got like 2 minutes. Yeah, there's some questions. You should have 2 minutes for questions, hopefully. Ah, yes, yours is the one with the video.
Okay.
Yeah, you can just check it. You have 2 minutes if you want.
Okay. The video?
Yeah, I have the video, yeah. This one. Yeah. Where is the mouse?
No, it doesn't have a mouse. You can use this.
And how can I start it? It's blocked.
What are you expecting?
It doesn't work.
Okay, to move.
It doesn't work. It's not working, man. It's blocked.
What is blocked? No. Is it the video that you want to play?
Yeah. It's in bed. It's in bed.
Okay.
But where is the mouse? Can I have the mouse there?
Okay. The mouse is not attached. It's not attached.
Yesterday it was working. So yesterday when I tried it, it was working on the computer, on the room 10. So you have to open it, or can I open it?
You can. And I could as well.
But the problem is that can I have the So can we open it with Adobe?
And then Control L. Okay. Cool.
But wait.
Okay.
And then what is the mouse there? What is the mouse?
Stop play.
Huh? What is the mouse?
Okay.
Okay, so it's 11 o'clock, let's get started. Very welcome to the community ecology session of Foodwebs and Trophic interactions. And our first speaker is Deirdre McLean, and she'll be talking about evolution of resistance to multiple enemies as shaped by biotic context. Thank you.
Cool. Okay. Thank you. So can you all hear me okay? If I don't stand by the microphone? Yeah? So thank you for coming. My name is Deirdre, and I'm a recently started postdoc in the University of Edinburgh. And today I'm going to talk a little bit about some of the work that I started during my PhD, and it's kind of been carrying on a little bit. So some of the data is quite new. We're still working on the mechanisms of how these things are happening, so I'd be really interested in any of your inputs. But I think some of the results are looking really promising, so I thought I'd show that to you today. So, yeah, I've entitled it The Evolution of Resistance to Multiple Enemies. And when I'm talking about the evolutions of resistance, what I'm talking about are defensive traits. So how species change their defensive traits depending on the communities that they find themselves in. So this is work that I did with my supervisor in Donoghue and Trinity, and Alan Finn as well, who has a poster later on, not related to this, but still, go check it out. And then Villa Freeman and Britt Koskela as well. And the ideas for this came around looking at communities as complex webs of interactions, particularly antagonistic interactions, such as predator-prey, competitive, parasitic interactions as well. And how do those interactions look from the point of view of one species within that community? So this was the motivation for doing this. So to a species in towards kind of the basal end of the food web, how does it view the rest of the community? And when you start thinking about that, you realize that most organisms spend their time wondering who's watching them. So is it a predator, for example? Is that what they're most afraid of? Are there parasites lurking in their environment ready to infect them? Or is it a member of your own species that might be sneaking up behind you as well? And species alter their responses depending on which of these they're dealing with. So they'll have different responses to predators, competitors, and parasites. So thinking about things like predatory interactions, defenses against predators might include external defenses, such as spines or an armor. You might try to grow too big for your predators to attack you, or there might be some complex behavioral interactions and camouflage as well, to return us to planet Earth and the iguana racer snake trauma. So the iguana here is trying to stand still to avoid detection from the racer snake. So there's a number of different mechanisms that species use to defend themselves against predators. Likewise then, thinking about parasites, there's external defenses, like the buffalo rubbing off ticks, for example, and of course immune responses as well. Competitive interactions then are a little bit maybe more tricky to predict. I think this picture beautifully sums up a lot of competitive interactions. So we've got two species of vulture fighting over a resource, so a carcass. And so the vultures are obviously different sizes, they've got different shaped bills, different lengths of necks, etc. And this divergence is largely driven by competitive interactions. So we know that they're a really strong force in structuring the community, but perhaps in a less predictive way than some of the other ones that I mentioned. And those responses to competitors will also depend whether you're competing against a member of your own species, where you're both equipped with the same toolbox and it's about how you employ that, or whether it's another species that you're competing against or interacting with. And like in this scenario, you're going to act differently depending on whether you're the bear and the wolf in that scenario. Okay? So that's kind of some of the antagonistic interactions that we're interested in. But thinking about how these kind of traits evolve, if you take even a simple two-species system, so predator-prey, and you think about some of the predictions that you might make based on this, so a three-legged iguana, for example, might not make it very far, it's going to be caught by the snakes. So you can predict some of the trajectories that this might go in. But even taking a two-component system, so this is one of my favorite papers of the year, and if you haven't seen it, I really recommend reading the paper, but particularly looking at the video. So these are the mega plate experiments that they did. And so this is basically a giant agar plate. And here this is a gradient of antibiotics, it gets stronger towards the middle. And what they did was they inoculated each side with a couple of colonies of bacteria and just allowed them to evolve resistance. But the cool thing here is these colored dots just show the track, basically the phylogenetic track that these species took. So even a very simple two-component system with one species can show really complex patterns. So what happens when we add more species on top of this? And this is what we were interested in. So to do that, I used bacterial microcosms, or microcosm communities. These are a lot easier to do these things in the lab, and you can track some more evolutionary responses much easier than you can with other species. But the really cool thing as well is the bacteria show a lot of analogous responses to the kind of antagonistic interactions that we were interested in. So anti-predator responses, things like biofilm formation, which is essentially an external response or defense. They show lots of anti-parasite mechanisms against bacteriophages, both internal and external. And there's a whole suite of competitive interactions that they employ as well. So this one here is a really cool one where bacteria employ a kind of jousting mechanism at each other. So they inject each other with toxins and try to suss out who's the stronger competitor in the scenario. And what we were interested in doing was taking a focal species, so bacteria in this case, and looking at how their responses to predators in the form of protists, parasites in the form of bacteriophage, and competitors as well, so another bacteria species, and how they changed in response to each of these, defending themselves against each of these three enemies. So we might make predictions on how they would respond to any of these pressures in isolation. So there's lots of different things that they might do, but what happens when all three are together at once, or they're present in any kind of combination? So for these experiments, what we did was we took our focal species, which is Bacillus subtilis, it's a kind of ubiquitous water and soil bacteria, and we grew it in microcosms with either of a competitor, a predator, or a parasite, and all combinations thereof. We allowed them to grow for 10 days without interfering too much. We did take some samples, And at the end of the experiment, what we did then was we isolated our focal species again. And because they're bacteria and they're really cool, you can freeze all these samples. So we were able to compete our focal species, so our evolved species, against the ancestral form of the phage, the predator, and the competitor. So we were able to see then how much they changed their defense mechanisms depending on which community they had grown up in for the last 10 days. So I'm just going to show you some of the results that we got from this. And there are a couple of predictions that we made. So two of them, one of the more obvious ones is that the more antagonists that you're present with, the lower your density might be. And the lower the density, the less likely certain mutations are likely to occur. Okay, so that was one idea. The other one we were interested in then was, would we see some trait correlations? So if you were very good at defending against a predator, does that also make you a really good competitor, for example? So this is just a sort of summary of the kind of interactions that we had in our system, where we have our two bacteria species that compete for resources. There was a predator which fed on both of these bacteria, not preferentially from what we could tell. And there was also a bacteriophage as well, which specialized on the one species of bacteria, our focal species. So as far as the results are concerned, then the first thing we were interested in was, well, has the biomass actually changed? Do we see a difference in biomass? And the answer is no. So we didn't really see much variation at all as far as the biomass of our focal species was concerned across any of our treatments. So this led us to think, okay, well, if the biomass isn't changing, if they're not doing much different ecologically, maybe there aren't too many evolutionary things happening either. Maybe the species are all kind of doing the same thing. But interestingly enough, and so we were kind of disappointed when we saw that. But then we started to look at some of the evolutionary responses. So this is when we took the focal species out and we were competing it against, in this case, it's our ancestral predator. Okay? Resistance here on the y-axis, the higher up on this graph it is, the more resistant it is to the given threat. Okay? So this scenario was where we've had our bacteria growing up with a predator for the last 10 days. So we kind of expected that if it's been pre-exposed to a predator, it's going to be better at defending itself against its ancestral predator. So we thought that this might be the highest one that we saw. But that wasn't actually the case. There's a lot of variation going on here. And we're still kind of trying to work out some of the patterns that we might see here. So a more obvious one came when we started to look at parasite defense. So this is how the bacteria defended itself against the phage. And here, so again, resistance, it's the same graph here. Higher up here is the more resistant. And what we found is when the bacteria had been previously exposed to a parasite, its response to the ancestral parasite was hugely varied. Okay? And when we look at the rest of the treatments then, one of the most interesting patterns to pop out is that when a competitor was present, the bacteria was a lot more resistant to the parasitic phage than it was when the competitor was absent but a predator was present. Okay? So something about having a competitor present made you much better, or made them much better at defending against the bacteriophages. Next then, we looked at competitive ability. And so again, resistance, the higher up, the more resistant it is. And this was measured as the ratio of our focal species to its competitor at the end. And what we found was, things missing here, but this is in the scenario where the bacteria had just co-evolved with that one competitor species. There was no other species present. And what we found was actually, quite strikingly, that was the only thing that affected competitive ability. So past exposure to a competitor was the only thing to influence the competitive ability of the species at the end. And we didn't find an effect of a bacteriophage or a predator on this as well. So just putting all of that together then, as far as our focal species was concerned, it seems like the villain of the piece, as far as the dominant interaction is concerned, was our competitor species. And it had more of an influence than either of the other two, particularly as concerns parasite resistance. And what's also interesting is that while having the competitor present influenced the susceptibility of the bacteria to the phage, that didn't go both ways. So the presence of the phage parasite didn't seem to influence the competitive ability of our focal species. And so we're trying to look at some of the mechanisms behind that now. So I've got some growth rate data coming in and things to see why that might be. So just to kind of sum it all up together then, again, when we're looking at our whole communities, and we think about the kinds of interactions in there, I think what these results are indicating, first of all, is the strength that competitive interactions might have on influencing some of the other interactions within species. But also, if we add or remove species from that environment, so if we change the biodiversity that's in there, we might not necessarily see a kind of ecological signal in the sense of biomass. If we go out and measure biomass, we might not see some of the evolutionary signals that are occurring in those communities, particularly as far as things like parasite resistance might go with other species in the environment. So watch this space for more, and thank you for listening.
Okay, we have 2 or 3 minutes for questions.
Yeah.
Can you just clarify, I didn't quite catch it. Did you find that the focal species, the bacteria, the biomass of that was not affected by the presence of paramecia?
Yeah, so we found basically there wasn't variation. So it's not, when it was growing on its own, so I don't have that data up there, it's higher than when any of the others are present. But, so there was a reduction, so it wasn't completely unaffected, but we didn't find it varied in response to the others. So yes, I actually should put the others, but yeah. So. And we also have the ancestral one as well. And again, the ancestor grew higher than the evolved one.
Could you comment on the experimental setup and environmental conditions you used? Was this on agar or in liquid form?
It was liquid, so sorry, I should mention that. So it was an aquatic microcosm setup. So it's basically just water with protozoan pellets, so a kind of nutrient environment in there. And they had glass microbeads in there as well to add some spatial structure. But they weren't shaken, so they were left room temperature for that amount of time. And then the evolution experiments were conducted in well plates in the same medium.
Okay, any more questions? Okay, when we have some time, we'll just move on to the next person. Okay, I'll introduce the second speaker. We have Samuel North, who'll be talking about integrating next generation sequencing technology into Foodweb ecology.
Okay, so can people hear me if I stand here? Yeah? Because I think it's too loud for the microphone. I'm going to talk to you about integrating next generation sequencing into Foodweb ecology. My name's Sam North. I'm a second year PhD student at Imperial College London based at Silwood. And my sort of secondary title is Ecologists Do Wear Lab Coats. So just a bit of background on the title. When I was first starting my PhD, I started reading Foodwebs by Pim. And I came across this in the introduction. I thought it was quite amusing based on what I was sort of tasked to do, because this is now changing in Foodweb ecology. So just a little bit of background to Foodwebs, why study them? So Foodwebs are an incredibly useful tool for studying a whole range of ecological issues. They allow you to understand ecosystem function, complexity versus stability, the link community and ecosystem ecology, but fundamentally, they allow you to describe the mechanisms behind species interactions, both directly and indirectly. And to me, they represent a real kind of core of ecology in this way. And this is very useful because they allow you to predict a whole range of different consequences of environmental perturbations, how ecosystem services are affected by agricultural activity, the effects of climate change and hierarchic ecosystems, because you can see the effects on individual species, indirect interactions, and the whole ecosystem and community on its own. So, but the problem with this is Foodwebs are incredibly complex. They require a vast amount of data to be empirically characterized, and this is exacerbated by the fact that traditional methods, such as gut content analysis, microscopy, gut content analysis, or actually experiments where you look at feeding interactions or direct visual observations in the wild, they're very slow and they're very labor intensive. Best case scenario, microscopy gut content analysis, which is what I'm looking at, you'll get things like this. So you can clearly see that these are both insects here. I'm not sure, can I get this to work? Yep, not sure about that. There we go. Where you can clearly see they're intact individuals, they're both insects. If you're familiar with them, you can tell they're both Propopteran. The genitalia is still intact, so you can get them to species level. However, more often, okay, not sure why that's happening. More often, am I doing something wrong here? I think it's... There we go. Sorry about that. Yeah, so more often, they look like this, where you can see it's just a sort of mess. A mess of kind of, basically body parts. Sometimes you'll get pieces like this, but it's very difficult to actually quantify and identify what you're looking at. Many guts are also empty, and really we need better methods of characterizing the interactions. Which brings me on to next-generation sequencing. So applying next-generation sequencing technologies allow you to identify prey items in predator guts through the DNA. Why do this? This has got several advantages. So first of all, you can get the so-called Russian nesting doll effect, where you're identifying secondary and sometimes tertiary predation of prey items in predator guts. They allow you a better bang for your buck, where you're getting more feeding links identified from trace amounts of DNA. And this all equates to more links per predator analyzed, which means that you don't have to identify as many gut content samples, which means it's less labor intensive. So the system that we worked on, the Broadstone, is a stream in southeast England. It's a very famous food web in Foodweb ecology, and it represents one of the best characterized food webs for any system to date. It's easily the best characterized one in Europe. It's got almost 5 decades now of food web research on it. So my PhD supervisor, Guy Woodward, did his PhD on this system, and his supervisor worked on this in the 70s and the 80s. And this means there's a lot of large historical data, a lot of information on it, which makes it the ideal candidate to test these technologies with. So we chose a subset from the macroinvertebrate food web of predators based on their position in the food web, their sort of how common they were, but also we included Betsia here, which is a suctoral feeding predator. So another advantage of using DNA-based methods is you're able to characterize the diets of liquid feeding predators, which you can't do using traditional techniques, or you can't do using sort of microscopic gut content analysis. So this was the experimental design. So we sampled in around spring 2015 using standard kick sampling techniques. These were then sorted on site into DNA and RNA-free microcentrifuge tubes and stored in 98% ethanol. And then 100 individuals of each species, 200 individuals with each species was collected and split in half. So 100 was done using the DNA pathway and 100 the traditional pathway. So with the DNA pathway, which is what we're more interested in, DNA was extracted, pooled, amplified using PCR technique, visualized for success, and we're currently at the Illumina MySeq sequencing stage. So we were just awarded a grant by NBAF to do our sequencing and our bioinformatic analysis, which is where we'll then scan the MySeq information we get back. We'll scan it using the Bold System online database to establish links. The traditional pathway we're using dissecting, so we've dissected guts, mounted slides, and now we're at the microscopy analysis stage. Just a little bit more on the DNA metabarcoding. So we were targeting the 650 base pair cytochrome C oxidase gene, which is the standard one to do for animals because it allows for a greater than 97% success rate of most animal groups. But because we were expecting the DNA to be quite degraded, we used so-called mini barcode primers, which amplify the first 120 to 150 base pairs, which allow sort of basically when you've got much more DNA, degraded DNA, it basically will anneal to the first sort of base pairs, and it's basically allows us to get more information out of it. And as I've mentioned, we'll be using the Bold System barcodes. And this will allow us to get all of these links in the macro and myothermal invertebrates. So you can see I've highlighted the invertebrates we're sequencing there. But this will allow us to get a huge range of it, and essentially it just leaves out the non-animal basal resources. And we'll be measuring technique effectiveness using yield effort curves, where it's the number of predatory guts examined versus the number of links. And you can see it takes several guts. So these are the traditional ones, traditional techniques using microscopic gut content analysis. And as you can see, it takes several hundred predators to be identified before the links plateau. And hypothetically, we expect to see something like this, where you're seeing a much higher asymptote because you're getting more interactions identified that previously weren't available. And you're also getting the asymptote reaching it much more quickly because of these identification from secondary predation and also from more unidentifiable parts. However, this is all in theory. The sequencing hasn't yet been undertaken, and the primers that we're using weren't specifically designed for this. So we're using primers that were used for targeting degraded DNA in museum samples, but they haven't been applied to gut content analysis before. And success is unknown until we'll get the sequence data back. And additionally, optimization of the parameters is very time consuming. So it took a long, long time to get successful amplification to happen. A lot of PCRs, a lot of electrophoresis gels. It takes a lot of time, which means more time and more money. Which brings me on to the limitations of next generation sequencing. So they require specialist laboratory and skills. They're very prone to contamination and inhibition, as I've already said. And they only allow for binary presence-absent links to be identified. Comparatively, they're also very expensive. And this, I think, limits its accessibility to traditional ecologists, which brings me on to the traditional ecologists don't wear lab coats. And whether or not you should use these techniques, I think, also, there's a very long period of time required before you actually get data. So unlike traditional techniques, where the more time you spend, the more data you get, with this, the more time you spend, you can spend as much time as possible, but the optimization stage takes so long, so you'll eventually get all the data at once. So it is still quite labor intensive. So whether or not you should use these depends on a whole range of issues, such as what sort of data are you looking for? So do you want quantified data, or will presence-absence suffice? Do you need detailed population data? A very important thing is whether or not the food web is already established, or whether or not you need to do this from scratch. Whether or not you're looking at a single species or a whole network, or a particular subset. And what facilities do you have? Do you need to design the primers yourself? Are the barcodes already available? Do you need to do the sequencing? And I think all of this comes together quite nicely, and I've tried to show how interconnected these different things are to make it look a bit more complex. But it depends on what your research question is. So this is a real kind of, I think, quite a sort of sledgehammer technique. So if you don't need it, if you're just looking at a single species, there are a whole range of other more efficient techniques that you could use. And I think this is probably best used as a suite of approaches towards depending on what your research question is. So, are they the future of Foodweb ecology? Yes and no. So in systems like the Broadstone, where they are extensively studied, they're looking at the real questions in Foodweb ecology, trying to answer a lot of the theoretical problems. They're the next step. This is, there's not really much more you can do with traditional techniques. But the success is still unknown. So as I say, I don't have any data yet. So I don't know, talk to me in 6 months, I guess. Other DNA-based techniques are available, which I don't have enough time to talk about now, but I've tried a few of them before, such as diagnostic PCR technique. They're much quicker, much less labor-intensive, and they're more accessible for people who aren't familiar with these kind of technologies. But fundamentally, it depends on your research question. The next steps of the project, just quickly, to integrate the non-animal basal resources and sequence the remainder of the consumers so that we can get the whole network approach, and hopefully we'll get an even bigger and more detailed network, and sequence the remaining consumers. And for other parts of my PhD, I'm quite interested in looking at other systems and other alternative DNA-based techniques for characterizing interaction networks. Just quickly thank my acknowledgements. So BBSRC for funding my doctorate, NBAF and NERC for the grant they've awarded us, the University of Essex for putting up with me for so long, and helping me to do my lab work, BES for hosting me, all of you for attending my talk, and my supervisors and those that helped me. And these are just my take-home messages here, but thank you very much for listening.
Exciting talk. And I was surprised to see the sampling curves that actually separate for the diets. And until now, all I've seen were always curves that do not separate.
Okay.
And indeed, I would expect the sampling curves grow with the square root of samples taken. So setting the discussion which is true to the side, if it does not separate, if there is nothing like a present and an absent link, you just get rarer and rarer diets. Do you have an idea how to incorporate this into your research?
So that one was a sort of hypothetical, and I kind of made it to look different. I think you will see a higher kind of plateau because for example, those curves there were mainly just looking at sort of the macroinverted.
Are you sure you get plateau?
Are you sure it was plateau?
Yes, it will plateau.
Maybe not with the numbers I'm using. So I expect we'll probably see a kind of an asymptote start to form, but because I'm using lower numbers, I'm only using the kind of hundreds around that area. I don't think I'm going to see them plateau that quickly. However, I think if you were to scale it up to 200, 300 individuals, you'd see it happen much more rapidly, much more quickly.
For fish, it doesn't happen.
Okay, well, I would have thought, for the stuff I've seen with macroinvertebrates, I would have thought it'd happen. But
So the secondary predation, how do you deal with that? Because I want to know the primary predation. At least one could mistake secondary and how does one separate secondary and primary?
well, that's one of the issues again with it. You can't, because you're just going to see that so you can't tell whether or not the predator has directly consumed it or if it's secondary predation. What I mean in that is that you're going to see more links appearing for this individual, but that is a limitation. So I think this is best used as a suite of approach. You need to know more about the system and the diets of the individuals. It's not a sort of silver bullet technique. It allows you to get a lot more links, but you can't separate. So it allows you to better understand, I guess. You could, like for example, say then you sequenced a predator. So say you get, I don't know, for example, Siphonoperla, one of the smaller macroinvertebrate predators in a Ecological gut. Then you sequence that separately, and you see there's a, say, high proportion of links for a feeding interaction that you're also seeing with Ecological. You could theorize that the reason it's showing up in Ecological is because it's secondary predation. But you have to know more about the food webs. It's not just going to build you a food web, basically. Yeah.
Nice. There's great potential in this sort of methods. But as you said, it's not alone, it's not enough.
Yeah.
So do you actually have estimates of the actual abundances of different species in this system?
Yes, we do. So we've got we've got a huge wealth of data. It's changing quite a lot at the moment. We're actually working on a paper looking at how, so a few years ago, about 10 plus years ago now, a trout species actually invaded the stream, and that's really changed the abundances. So we're actually looking at publishing a paper on the recent population dynamics, basically, and the numbers of individuals of different species, but we are seeing a shift. But yes, to answer your question, we do have a wealth of data, 5 decades of how it's changed, and we've actually got this really cool figure where you're seeing a trivariate food web from how it's changed through species invasions over 5 decades now.
That's going to improve the kind of the estimate of the robustness of your links between
Yeah.
the abundance of the different species.
Yeah.
Very quick time. We'll have time for one very quick question. No? Okay, then we will move on. Thank you very much.
Thank you very much.
Okay, our next speaker is Ulrich Rose, and he'll be talking about the scaling relationships of consumer and resource body masses across ecosystems and interaction points. Yep, thank you.
Thanks very much for the introduction. Thanks very much for showing up here. So we're going back in time. This is also Foodweb research, but these are some rather very traditional patterns. So this is going back the research idea of looking at the structure in terms of the body masses in the food webs. This is going back to the Newtonian idea that we have pyramids of numbers, biomasses, and masses. And with this, I would like to take you to the first slide where we define a bit what we're talking about here. So you all know what a body mass is. So the example I'm giving you here is a terrestrial one. So we talk about a predator that in this example is a 5-kilogram fox predator that feeds on smaller resources. This is like a 0.5 kilogram rabbit. So this is the body mass. This is the mass of an individual in this population. And in Foodweb ecology, we often normalize this to body mass ratios. So that means we express the mass of the consumer relative to the mass of the prey. And in this example, this means that we divide the 5 kilogram of the fox by the 0.5 kilogram of the prey, that is the rabbit. And the body mass ratio of this specific example is 10. That's unitless and means that the consumer or predator is 10 times larger than its resource. And this is independent of the resource and consumer masses, so this can be protists in their prey or vertebrates in their prey, as in this example. So this is the body mass ratio. And the third thing I'm going to use in this talk is the scaling relationship of body masses. It's also often used in food web ecology. One of the masses now is on the y-axis and the other one is on the x-axis. In this talk I choose to put the consumer on the y-axis and the resource on the x-axis, but this can be swapped. This has some implications that I'll tell you later, and there's also a reason why I chose to do it this way. So these are the three types of data or dependent variables I'm going to show you. It's masses, body mass ratios, and body mass scaling. Why is it important? I could go through a lot of slides. So through the last decades, a lot of research has been done on body masses, body mass ratios, and the scalings of them in food webs. The examples I'm going to give you here are for body mass ratios. So here, if you have three different consumer resource pairs, terrestrial ones, these are the ones that we have in the lab, and these are experiments on their feeding rates depending on prey density, and they are ordered that according to body mass ratios, the smallest body mass ratio is here, and the largest one is here. And you very quickly see if you go through this row here that the feeding rates change with the body mass ratios and that they go up. So body mass ratios have implications for the feeding rates and thereby for the interaction strengths of these specific links. Interaction strengths have all kinds of implications in food webs. One of them is that they change the population dynamics. So for the same three species here in this example, we have simulated population dynamics of the densities through time, and you see that in this case that the population dynamics change. They can change in several ways, so there is not a unit way, but there is one further implication. Now we go back deep in time. If we look at body mass ratios and create model food webs and create food webs with different body mass ratios, so here this is the log 10 on the x-axis. The 0 line now means that consumers and resources are equally sized. Everything higher means that the consumers are larger. Everything smaller implies that the resources are larger. For instance, the 2 means a body mass ratio of 10 to the power of 2, that the consumers are 100 times larger than their prey in these food webs. Only theory, but it indicates that as soon as we have higher body mass ratio, the stability of the food web is higher compared to food webs with lower body mass ratios. Stability here is the persistence probability. That's the probability of the species in the food web to survive and not go extinct. Just one of many measures of stability. Hopefully we won't discuss this after the talk, but I see some experts here. And just one measure. But others are also related to body mass ratio. It's a very good structural measure indicating or having secondary implications for stability. So this is why we are interested in food web research in body mass ratios. So we know a lot about body mass ratios, but there are some things that we still addressing. So we know a bit about how body mass ratios vary across different groups of species or interaction types. And one of the questions that we're interested in is which in any system, in any food web, we have different types of interactions. They are mixed. This is not pure theory. In empirical systems, we have small body mass ratios, large body mass ratios in the same system. We have the idea that the large body mass ratio interactions stabilize the systems, and we would like to find out which of the many interactions in natural ecosystems possess this characteristic that they have large body mass ratios. And so traditionally we asked or compared different interaction types or different ecosystem types. There were some studies that also looked at differences across metabolic types, so predominantly comparing invertebrates to a different group of vertebrates. But what we're also interested in is to look at other traits. And here, as an example, in this talk, I'm going to show you some data on different movement types. So in many food webs, we have species that have different movement strategy. They fly, they swim, they walk, although they don't move at all. So they're sessile. And these consumer types have different body mass ratios. So this is the core question of the next slides. what is the difference in these body mass ratios across these things? To answer this question, we started pulling together a new Foodweb database, so we needed more data. These questions are very data hungry, so we pulled together a new Foodweb database. Honestly, the talk probably has like 50 co-authors, so I skipped this with an et al. In the beginning. So there's a lot of people here, including the ones from Guy Woodward's group that contributed towards the stream. So one of the circles you see here in the UK for the stream Foodwebs is Broadston Stream, but there's many other Foodwebs too that we started to pull together. And we have different ecosystem types, as you see here. They're not as evenly distributed as I wished for across the globe, but it's a good start. And so right now the status of this still unpublished database is that it has 209,000 links of consumers and their resources, so it's about an order of magnitude bigger than anything we had before. It has at least information on the body masses of the consumers and the resources, but it has many additional traits as columns in this database. So the idea is that we pull this together, and at some point we make this publicly available for everybody who's interested in comparing different systems. So the first thing is that we usually look at the body mass ratios for different interaction types. So here this goes from bacteriivores interactions to predaceous interactions, and you see this is confirming existing knowledge that, for instance, for bacteriivores interactions we have very high body mass ratios, and for instance for parasitic and parasitoid interactions with their host they have very low body mass ratio. Not a big surprise, but there is quite some variance in the predaceous interactions, so there's a lot of variation that is not explained by the fact that they are simple predaceous. And one thing that we separated within the predaceous interaction, there's very clearly two groups of data and different strategies, so we separated individual-based and non-individual-based interactions. So the individual-based interaction is that an individual consumer hunts and targets a single individual of a resource and overwhelms it and consumes it, whereas non-individual-based or interactions are that consumers target groups of resources, swarms, flocks, etc. And typical examples are filter feeding or grazing consumers. And you see that the non-individual or needy consumers have much higher body mass ratios than the ED or individual-based interaction predators. But still, there's a lot of variation in here. So we're zooming into the database, and for the next slide, we just look at the individual-based interaction predators. Now we switch. We go to the body mass scaling, consumer mass against resource mass, and here we have a one-to-one line. That's not a point now in these diagrams, but it's a dashed line where they are equally sized. And high body mass ratio applies now that you go in orthogonal direction upwards from this line, and low body mass ratios are lower than this line. So this is the swap you have to make. Okay. I know this is challenging, but these are the data. So first thing, traditionally plotted here, consumer mass against resource mass. Different colors are different ecosystem types. So here are the lakes, marine ecosystems, streams, and different terrestrial ecosystems. You probably can barely read this, but green is terrestrial below ground, and pink here is terrestrial above ground. I see positive scaling in all cases. There are some specificities. So the regression lines here are standardized major axis regressions. This is because you can exchange the x and y axis, not typical x-y relationship. So this is why it's standardized major axis regressions. And you see here that we have, if we go to the ecosystem types, consistently high body mass ratios for lakes and very steep scaling relationships for streams, but very shallow and low relationships for terrestrial systems. So many aquatic ecosystems are characterized per se by high body mass ratios, whereas terrestrial systems possess many low body mass ratios. Not terribly surprising, just shown with new data. And we also zoomed into the different metabolic types of the consumers. So we have here ectotherm vertebrates in green, endotherm vertebrates in orange, and the invertebrates in purple. And you see also different scaling relationships. The high body mass ratio, the increase here is driven by the pooling of the different groups. So within groups we have relationships where the regression lines approach the one-to-one line. So within groups the highest body mass ratios are caused by interactions between the smaller species, but when you pool them then you have a positive relationship. And the last result is for one of the traits that we have in the database. It's for movement types. So here we separate swimming, walking, flying, and sessile consumers. And what we see is that flying consumers and swimming consumers per se have higher body mass ratios than the walking or sessile consumers. A lot of data, just to quickly wrap this up. So the high body mass ratios, just to go back to the start of the talks, are the ones that we think stabilize food webs. They are here highlighted in light blue, probably not really visible as I see right now, but here they occur across ecosystems. They occur definitely in lakes, a bit less in marine ecosystems, but they're much lower in terrestrial food webs. High ratios occurred for vertebrate consumers, but not for invertebrate consumers, and they occur for flying and swimming predators, but very low for walking and sessile predators. In conclusion, this means that there is a chance that stability in these systems is mainly occurs in aquatic ecosystems, but then in the other systems is mainly driven by vertebrates, and in particular if they're flying or swimming, to make it really, really simple in the end. Nature, as always, is more complex, but this is the way to get a first glance at it. There's so many people to thank. I'll cut it really short. There's so many of even you here in the group that supply data, which I'm very thankful for. This is a project of the entire working group, Econet Lab Active, so we all contributed with data analysis and ideas. And also thank you very much for your attention and coping with the body mass ratios and scaling stuff. Thanks.
Time for one quick question.
Did you try to include parasites as well?
Yeah, initially yes, we separate them, so we don't analyze them together with predaceous interactions, so we separate them right away, because the first split that we do is amongst the interaction types. So here I just showed the analysis for predaceous predators, but you can also look at parasites.
Anyone else have a burning question? No? Okay, thank you very much. Here we go. We have a video in this, so it's going to take a little time.
You should just have to drag it over to the other screen. It should just play automatically.
Can we try?
Do you want to try it now?
Yeah.
So you just have to get this window, drag it over, and then full screen. Is that okay? So you'll have to come out of the PDF first. You've got to close that one. Is that okay?
Okay, that's great. And then go through.
Yeah.
Okay, here we are. So hello, I'm Gian Alberto, and it's really a pleasure for me to present a chapter of my thesis that I'm carrying out in Zurich. And I'm talking about networks still, but we are jumping into the amazing world of plants. So a long-standing question in biodiversity, in ecology and evolution is why are there so many species around? And several theories have been proposed. And as we have seen so far in these days, competition and biotic interactions seems to be a very important mechanism determining the coexistence of many species. However, we also do know that positive interactions are very important. We have seen from Benno the importance of pollinators, and there is increasing evidence that positive symbiotic interactions, as mutualism, are relevant for species biodiversity. And but what about the network of interactions? Because research in plant community has been focused mainly in pairwise interactions. However, we know that, as we have been seeing, that species are embedded in a complex network of interactions. So the goal of this chapter is to develop tools and to understand the structure of the networks between among species, and in particular what is causing these network structures and how it is changing with scale. So we are basically kind of putting our network beneath a microscope and zooming and checking how the structure is changing. In particular, I will respond to the following questions. So, to define the scale at which interactions between plants are taking place and how the structure of the network is changing. The methods that I'm developing starts from sampling and mapping the community. So we went out in the field, in a fragmented tundra ecosystem, and we established a grid, 3 meters per 9, and for each individual plant, we record the species identity and the coordinates. After this, we run for each species the universe distribution that is mainly stochastic or niche determined. In other words, we check whether some neutral process like random dispersal or mortality can determine the distribution of the species, or whether other typical niche model like the soil richness can better explain the distribution of one species. After this, we develop models to account for the bivariate species distribution, that is to check whether accounting for the own species distribution, we can see still some dependencies among plants. And in particular, this dependency can be positive, can be negative, or can be neutral. Given this bivariate distribution, then we built our network, starting from the adjacency matrix that was represented so by the plants as node, as we can see here, and the links between plants were positive, whether we observe a significant positive deviation for what we would expect by chance, negative in case there was a negative correlation between the two species, or neutral elsewhere. So let's see if the video is working. I hope so. Sorry. So basically, what we will see is this network that we put under the microscope. So I'm zooming from a very, very small, fine spatial scale to a larger spatial scale. So positive interactions are depicted in red, and negative interactions are depicted in blue. Okay, thank you very much. That's very kind. So as we can see in the beginning, the network is quite dynamic, I would say, first of all. And we see that there are lots of positive interactions, and the network is quite cohesive. And with increasing spatial scale, we can see that the network starts to fall apart, and negative blue interactions are becoming dominant. Can you listen to the music? Yeah. So, and basically in quantitative terms, what we observe is that positive interactions decrease with increasing scale, while negative interactions are increasing. And we were able to identify a shift in the predominance of interaction type at about 25 centimeters. So we have that till 25 centimeters positive interactions are prevailing, and then after roughly half a meter, negative interactions became predominant. But what about also if these interactions were reciprocal? In particular, we observe that positive interactions, that is, a plant that is interacting with another plant, also receive a positive interaction, a benefit. They decrease quite fast, and then they tend to disappear. However, positive neutral interactions remain steadily decrease. Considering negative interactions, we have an increase in both reciprocal and non-reciprocal. Interestingly, we didn't observe any positive to negative interactions. That is, in general, if there is a positive interactions, the other plants respond with a positive or a neutral one. If they are negative, they do the same, but we do not observe a positive interaction that is counterbalanced by a negative one. Looking at the effect of positive interactions on the x-axis, on the richness of interacting species on the y, we observe a quite nice relationship where with the increasing of positive interactions, we have an increase of the number of interacting species. Then considering the network structure, as we have seen, we can nowadays analyze the network in lots of different ways. What I found interesting is this property that tomorrow Santiago and Stefano will talk about more in detail. But here briefly, I consider transitivity using a metaphor from social networks. So basically, if you consider your two friends, this network is transitive whether your two friends are their friends as well. In this case, if that's me, those are the two friends of mine. If the two friends of mine are not friends as well, this approach to intransitivity consider this network intransitive. And that's the interesting result. We observe a strong decrease in transitivity, that is an increase in intransitivity, and at one point the system became completely intransitive. And but we observe that this high transitivity, that is usually quite rare in nature, but also in other complex networks like social networks, it's strongly related to the presence of positive interactions. So it means that positive interactions can increase the transitivity. Then I consider another property, that is the size of the largest connected component, that can give us an idea about how the network is robust to changing and it's stable. In particular, we quantify, it's a very easy and straightforward metric. In particular, it is quantified by just the number of nodes that are connected with considering the biggest part of the network. Or in other words, in this case, we have 5 nodes and all of them are connected, so the size of the largest connected component is 5. In this case, we still have 5 nodes, but the network is falling apart, so we have one component composed by 3 nodes and the other one composed by 2. So the size of this network, the size of the largest connected component of this network is 3. And what we observe, as you might have appreciated from the video, is that with increasing scale, the network starts to fall apart, or what we call in more technical terms, there is a network breakdown. And in particular, we observe that at about 50 centimeters, the network reached the lowest size of the largest connected component, and this is strongly related to the competitive interactions. So in other terms, competitive interactions lead the network to break down. So stepping back to my original questions, with this method, we were able to clearly define at which spatial scale interactions are taking place. In particular, positive interactions are acting up to 25 centimeters, and negative interactions, they start from 50 centimeters. And we also do observe that intransitivity increase with competition, or facilitation increase the transitivity, and we observe a network breakdown at higher spatial scale. So this is the take-home message. So we observe at fine scale, positive interactions are prevailing, the network is robust and complex enough to resist to eventual perturbations, and with increasing scale, we have an increase in competitive interactions and the network breakdown. Sorry. Then, okay, I would like to thank my advisors, my colleagues, and who is funding my study and my traveling here, and thanks to you for your attention.
So you showed that there's a switch from positive to negative. But what it didn't show is are the same species switching their relationship, or are that those are dropped as you have some other negative relation with some other?
Yeah, so most of the species they keep the relationship. So the one that are usually benefactor, they keep to provide these positive interactions, and the one that are usually more competitors, they remain competitors. Yeah. Yeah.
You're working in a tundra system where there's lots of empty space. Most of us work in systems where the plants are all cheap by jowl. Does that change the relative importance of these sorts of competitive versus mutualistic interactions?
Yeah, definitely. That's a good question. We went in this tundra ecosystem where we know by literature that the positive plant interactions are relevant for biodiversity. Yeah.
Okay, thank you very much for your time. Okay, our next speaker is Tom Ames, talking about zooming into plant flower visitor networks using an individual trait-based approach.
No, in that one. Okay, well, I won't reiterate my title. So basically, I'm going to talk to you. You can hear me at the back, okay? Can you save me going through the microphone? So I'm just trying to take a slightly different approach to examining mutualistic interactions. And I'm zooming in like the previous speaker, but I'm actually doing it in a slightly different way. So I probably don't need to remind you, but the traditional way of constructing networks is focusing on creating nodes that represent species. But the problem with this is that the interactions that we're actually trying to characterize are occurring between individuals within the community. So perhaps we're missing important information by this sort of lumping into species-based nodes. So there has been some work done on trying to shift the focus into individuals and looking at individual traits in aquatic food webs. I'm not going to talk about that now, but it's really quite interesting work showing that by incorporating functional traits such as body size at the individual level, you can increase predictive power of some of the network approaches. And it would be really nice to try and allow us to do better cross comparisons across different systems, network types, by having similar sort of functional traits that we might be able to compare. Ooh, I've just done end, haven't I? So it's all right, I'll revert it again. So the aim of this work that I did really was to try and come up with a new approach for studying mutualistic networks, and in particular trying to compare the sort of traditional species-based approach with an individual trait-based approach. And so to try and address this, I just did a sort of bog standard bee transect. So I forgot to say that I'm focusing on bees as my mutualist bee-plant interactions, just to try and simplify the system. So collected some interaction data, but rather than just recording what species are interacting with each other, we were actually capturing the individual bees and measuring functional traits, which I'll come to in a minute, and likewise recording the functional traits of the flowers that they're actually interacting with. So for the bees, we all know that tongue length is an important functional trait in terms of determining how bees interact with plants. Difficult thing to measure with lots of individuals in the field. So we actually had a look to see if there's a simpler measure. Obviously body size would be great, and actually a simple measure of body size is the intertegular distance, so basically the thorax size. And we found in our community there is quite a strong correlation between tongue length and intertegular distance. And this has been supported by some more recent work that's been published on a wider variety of bee species showing similar patterns. The advantage of intertegular distance over tongue length is it also captures things like potential flight ranges and also relates to energetics, but not something that we've tackled particularly at this stage, but it's something to consider for future. So in addition to size as a functional trait, we were thinking, obviously when bees or other flower visitors are visiting flowers, they're actually doing different things. So are they just collecting nectar like this Bombus lapidaris here, or just collecting pollen like this Laser-glossum here? Others cheat, they bite into the flowers and steal the nectar that way. So what influence does this behaviour have on the sort of configuration of interactions within these networks? And for plants, we went again with sort of trying to go for a size-based functional trait. It's widely recognised that nectar holder depth is important in terms of determining what insect visitors can interact with that plant. Again, difficult thing to go and measure lots of individual flowers, especially when you've got composite flowers where you've got multiple florets to measure. So we, again, had a look to see if there's a simpler way of measuring or estimating nectar holder depth at the individual level. And there's a bit of noise in the data, it varies across species, but even just a simple measure as the maximum floral display size, so really just the diameter of the flowers, the size of the flower head, is a good predictor of the nectar holder depth. Obviously, you need to look at different relationships between the different species. So we could just simply go out and measure this, the size of the flower, and then predict how deep the nectar holder was for that plant. So in the aquatic foodwebs, the trait-based approach has generated sort of bins of size bins, and that's been done trying to produce sort of evenly distributed size bins. Problem with that is you often get empty bins, and it gets a bit messy when you're trying to look at the network. And we all thought it would be quite nice to try a slightly less biased approach and actually use a cluster analysis technique to try and to actually really look at what the variation in traits there is and try and cluster those using sort of standard cluster analysis algorithms. So we use an agglomerative approach, so starting with all individuals being represented by nodes and then sort of clustering them based on similarity in their traits. So we took an approach where we did a size-only approach, so the nodes are based on purely individual size, and one that involved individual size and behaviour. And obviously, to be able to compare these with the species-based network, we needed to make sure that we constrain the number of nodes to match the number of nodes in the species network for proper comparison, so we constrained it to the same number of species. But we also thought then it'd be quite nice to actually take it further and actually take that constraint away and just see how the approach would work if you just actually let the cluster analysis optimize the number of trait nodes in the network. Okay, so doing the thing that you shouldn't do is put a horrible complicated, or not complicated, but a busy table. Really, it's just a simple take-home message here. So this is just for the size-based network because it's a bit easier to show. And this is for the traits of the plants, so based on nectar holder depth of plants. And what you can see is that you do get some clusters with multiple species in, and then you can get, say, the dominant species, which is Centaura nigra here, is actually split across a range of different nodes. So you're sort of breaking down that sort of lumping all individuals of a species together. And you see the similar pattern when you look at the bees. So this is the size of the bees. I've removed quite a few rows just because there's a lot more bees than plants, which is quite normal. And again, you can see much more strongly that you're lumping multiple species into trait nodes. And again, you're splitting, some species are being split across several nodes. Again, quite different to what you would see if you just thought about it in terms of species. Okay, so what do they look like? I'm not going to probably dwell on the way that you plot these. I'm sure hopefully most of you are quite familiar with this. So it's, again, something you don't want to look at the fine detail, but it's just a representation of the bipartite networks. But what we've done here is we've ordered all of these by size. You've got plants along the bottom, bees along the top, and the boxes here are ordered in increasing size from left to right. So we can see whether there's any size structuring the network. Sort of hints about when you look at the species level, but as soon as you shift it to looking at size-based traits, you can see that indeed there is quite strong structuring with the small bees interacting with flowers of shallow nectar holders, and shifting up to large bees going for the deeper flowers. You also can see, which we'll pick up on the next slide, is that there's quite a lot more even distribution of linkages within this network using the traits. It gets a little bit more complicated when you introduce the behaviour into the network. It doesn't really impact too much on the size structuring, but you start to explain some possibly odd-looking patterns where you've got small bees interacting with large flowers, and they're often things like pollen-collecting bees, like that laser glossum that you saw earlier. Again, I'm not going to have time to dwell on all the detail here, but obviously that's just quick visualization. But what does it mean in terms of the classic metrics? So these are going back to the old metrics. It'd be quite interesting to try it with motifs, so it's quite a nice idea. So we just looked at the comparison between some of the sort of commonly used metrics that relate to robustness, stability, and so on. And what you can see is perhaps the most important thing that jumps out at you is there's a big difference in terms of weighted nestedness. So the trait-based networks seem to be much more nested, so they've got a much stronger hierarchical structure where you've got specialists interacting with species that are a subset of species being interacted with by the generalists. And there's plenty of evidence suggesting that this conveys greater robustness to extinctions in the network. We also see the trait-based networks are less specialised, so you've got less niche separation in there and potentially greater redundancy, which again we all know is potentially useful in terms of stabilising networks. And the other one worth pointing out is rather than just doing this traditional connectance, we looked at weighted connectance, so it's taking the linkage density of individual nodes into account there. And some recent work has alluded to the fact that it could be that there's a positive relationship between weighted connectance and stability of networks. And they've also used a slightly different approach to us. It's something that needs to be investigated further, but it suggests again that we're getting greater weighted connectance in these trait-based networks. So I guess the take-home message here is that there's a number of network metrics that we're finding, not huge differences, but sufficiently different when we take the trait-based approach, suggesting that maybe these networks might be a little bit more stable or robust than we would have thought if we just focused on at the species level. Then it gets quite interesting when we take the constraints away. Obviously, when we're looking at size, we've only got a single trait. I think if we introduce some more functional traits, potentially we might see a slightly different pattern. But if you actually allow the cluster analysis to optimize the number of traits, we can see they're greatly reduced in size. So we get much smaller networks in terms of trait nodes. And perhaps what was quite interesting is even introducing behavior into these, again, didn't really have any significant impact on the various structural parameters. So it shows that size seems to be the most important factor in determining the interactions in these networks. You do pick up some interesting patterns, though, by including behavior. Again, looking at, if we look here at this green box, that's female bees which are nectar robbing bees, and it shows that they're interacting with the largest flowers in the system. It's explaining why, and sometimes some of these might be a little bit smaller than the flowers that are interacting with. So it gives you new information, but probably just focusing on size alone gives you enough information to look at some interesting patterns. So really, in conclusion, I think it's quite an interesting approach, and I think for me, the important aspect is that we're not suggesting that you should take a trait-based approach in separately from the species-based approach. We shouldn't just ditch it because it's not very useful. It is useful, but by looking at it from the perspective of incorporating individual trait variation, it does actually reveal patterns in the network configuration that could
be important in terms of understanding stability, robustness, and so on. So it does give us some new insights by taking this approach. Obviously, we started with just a subset of mutualists in this particular system, so we really need to take it further and look at other pollinator groups. And I think there we might start to see some more interesting patterns in relation to modularity and so on at that point. And I also think the approach might help us out when we're trying to look at much more diverse systems. A lot of network theory is based on fairly simple systems just because of the logistics of measuring it. While it's logistically difficult measuring traits actually during the individual interactions, it might be possible to measure trait distributions and start to construct networks based on the trait distributions in quite complex systems where we might not have such good taxonomic knowledge. So I think it's quite a useful approach that's well worth exploring a bit further. I think that's it. I probably should have gone back to my previous slide. We skipped my acknowledgements. I just want to acknowledge the British Ecological Society funded this work, and Anglo Ruskin funded me to employ Beatrice Rameau, who's driven the network analysis for me on this as well. So, and thank you very much for your attention.
Some people say that you get the nested networks only because some bees have longer tongues and so can go deeper into the flowers.
Yeah.
And you should be able to see how much of the nested structure is explained by size structure.
That's a good point. Yeah, it's something we haven't really specifically focused on, but yeah, no, that's a really nice idea.
Just quantify it.
Yeah.
It's not yes or no, it's something
Sure.
between, but
Yeah.
nice to know.
I mean, obviously, the body size is important there in terms of whether they can get their heads in as well. So which is why not going for tongue length alone sort of captures slightly more. We thought that was perhaps a useful way of doing it, and it simplifies the measurements as well.
Okay, any more questions? No? Okay, thank you very much.
Thanks.
Yeah, next speaker is Callum McGregor, about construction, validation, and application of nocturnal pollen transport networks using molecular methods. Thank you.
Yeah, great, thanks very much. Yeah, thank you everyone for coming along. The work that I'm going to present today was really made possible by the ongoing collaborations between Darren Evans' group at Newcastle University and Darren's former colleague Dave Lunt at the University of Hull. And it's come out of this idea that I've been pursuing the whole way through my PhD that although most of the studies of pollination networks that we see in sessions like this focus on this kind of component of the network, which is mostly bees, hoverflies and butterflies, and maybe some other things like non-syrphid flies as well. There's a whole other component of pollination networks which we haven't really looked at in great detail, which is mostly moths, nocturnal pollinators. And my PhD has been looking at how important they are and how they're affected by various drivers of change. So to start out, we looked at the evidence so far for how important they are. And really what we found was a very surprisingly small number of studies when you consider the extraordinary diversity of those studies. So we had all sorts of different ecosystems, really wide geographic range, really wide taxonomic range when you look at the plants involved, but there weren't very many studies. What was also striking was that most of those studies were based on flower visitor observations. Now, we felt that there were problems with doing flower visitor observations for nocturnal moths, which are quite apparent, because to do flower visitor observations at night, our sight isn't very good. You really need to light the system. But moths respond behaviourally to artificial light. So you don't know if you're biasing your system by doing flower visitor observations. The real alternative to flower visitor observations is pollen transport studies. And we knew there were additional benefits to doing pollen transport. This study rather nicely showed the accumulation of interactions as you increase your sample size for both the F is flower visitor observations and the P is pollen transport. So you can see that you accumulate interactions much more quickly for the number of samples when you use pollen transport studies. And so we went about doing pollen transport studies, and that's what we used in our global change biology paper that was looking at the effects of artificial light on nocturnal pollination. And we found that to some extent they work quite well. And some pollen grains, when you get them under the microscope, they're really easy to identify. For instance, this is unambiguously honeysuckle, Lonicera, and this, in this case, Buddleia. However, that's not always the case. Sometimes your pollen grain might be obscured by other features on the microscope slide. Sometimes it might just be straight up really indistinct and have no real distinguishing features by which you can identify it. And so in the eventual study, we were looking a lot at quantity of pollen and diversity of pollen, but we weren't really able to look in detail at what species were forming the interactions, because in a lot of cases we just weren't sure. So we wondered if there was a better way, and we're not the first people to wonder that, and there are various studies already coming through showing that you can use DNA-based approaches to identify pollen that you've taken from pollinators. I've got two up here, and Andrew Lucas, who's in the room, presented a rather nice one with hoverflies yesterday as well. And in fact, this study even went so far as to compare metabarcoding to other methods of generating plant-pollinator interactions, and they looked at flower visitor observations, and sure enough, they found metabarcoding detects more interactions than flower visitor observations. But metabarcoding is a pollen transport method, so you might expect it to detect more interactions than flower visitor observations. What we wanted to know is, if you take the traditional approach of going at pollen transport, and you take this novel approach of going at pollen transport, how do they compare? So to that end, we went out, we sampled a load of moths using a blacklight Robinson trap that looks a bit like this on my balcony. We were working in an agricultural field margin in the northeast of England. Well, several agricultural field margins on a single farm in the northeast of England. It was in conjunction with another experiment looking at artificial light, but we controlled for that and checked statistically, and it didn't have any effect, so we don't need to worry about it any further. We then split the moths that we sampled in two, so that we had two twin samples. Each sample contained the same number of moths, the same set of moth species, in the same proportions. So obviously there's going to be, you can expect some stochastic variation in the network just by different individuals of the species interacting with different plants, but on the whole it ought to average out, because we have the same moths forming the network. One of those samples we went at with the traditional microscopy method, where you take a little blob of sticky gel with some Fuchsian stain in it, you swab the proboscis of the moth, you melt the gel onto a microscope slide, and then you identify and count the pollen under the microscope. The other sample, we developed a metabarcoding approach to detect and identify the DNA present in the pollen on the proboscis. I'm not going to go into great detail now for time reasons about that method, but if you're interested, come and talk to me, and I'm happy to go through it with you. And we had a number of hypotheses about what we expected to find when we compared these methods. We predicted that metabarcoding would be generally better at detecting pollen, that would detect pollen on more moths with greater diversity, and that we might resolve those pollen identifications down to lower taxonomic levels. We expected that to have an effect on the emergent properties of the networks that we constructed from the two data sets. And this is a bit of a hand-wavy hypothesis, but we expected to be surprised by some of the identifications, to look at identifications coming out from the metabarcoding network and go, yeah, I guess moths could be interacting with that, but we hadn't thought of it before. Broadly, that's pretty much what we found. So we found an increase in the number of moths on which we detected pollen with the metabarcoding method, which is the left-hand column. Interestingly, that didn't translate to any increase in the number of moth species that we found to be carrying pollen. However, we did find an increase in the number of plant, I've used the term operational taxonomic units, but I'm going to call this pollen types from now on. The number of types of pollen that we detected with metabarcoding was higher. And yes, we did identify more of those to species by quite some distance. More of the moths were found to be carrying more than one pollen species as well, which was interesting. And overall, the effect was that although we detected lots more unique interactions, so an individual moth interacting with a plant species, there was only a 20% increase in the number of unique interactions, so a moth species interacting with a plant species. So we're getting a picture of two networks that are pretty much the same network, but one of them's sampled a little more densely. The effect on interaction sampling completeness was quite striking. So the completeness of our metabarcoding network, despite sampling more unique interactions, more total interactions, was estimated to be much lower. Looking at some of these statistically, we found a significant increase in the rate at which we detected pollen on individual moths, including the rate at which we detected multiple pollen types, but there was no change in the rate at which we detected pollen on moth species. Similarly, the number of pollen types that we detected on average on individual moths was higher for metabarcoding than it was for microscopy, but there was no change in the number of interactions that each moth species formed according to each method. So again, we're getting this picture of sampling the same overall network, but sampling each of the links more densely with the metabarcoding approach. And that carries through into the actual network metrics that we calculated. So most of these structural networks are pretty similar for the two networks, really strikingly similar in some cases. There were two that I've highlighted that were notably different. Linkage density is essentially the number of links that each species forms in the network. That was higher for metabarcoding. Generality is the number of plants per pollinator in the network. That was also higher, although there was no change in vulnerability, which flips that round and looks at the number of pollinators per plant. Therefore, we think that both of these have come about because of the increase in the number of pollen types in the network that metabarcoding offered. I just want to talk briefly about some of the species that we detected and didn't detect with the metabarcoding method. Reassuringly, we detected some species that we kind of expected to be there, species that we know moths associate with, such as ivy. We also detected some species that surprised us, as predicted. But some of these are, in some cases, pretty plausible. We know clover is very abundant in the system. We know elder was there, and it's white, which makes it a candidate to be a moth flower. I've included this one because there's some fantastic and well-studied examples of tropical interactions between moths and orchids, but to my knowledge there's no interactions known in the UK between moths and orchids. Here's a hint that there might be some. There are also some crop species in there, which is really interesting. Again, there are no known interactions that I'm aware of between moths and crops in the UK. And we're not for a moment saying that this is proof that moths interact with any of these crops, but it's an interesting starting point for some more in-depth studies into things like single visit deposition. This is a way in which we can generate candidates for those more intensive studies to look at to see whether moths do contribute to the pollination of some of these crops. However, there were certain species that we were pretty certain were in the system, certain interactions that we were pretty certain were in the system that didn't appear in our final metabarcoding network. We have fairly good evidence that this is because the metabarcoding method isn't great at detecting things that are in very low quantities in wider samples of pollen. So if a species interacted and was carrying a lot of pollen of one species and one pollen grain of Silene might not be detected in sufficient quantities to end up in the final network and would be cut out by some of our contamination measures. Yeah, so there's that. I just want to spend a quick minute or so at the end talking about some of the potential applications of metabarcoding as we see them. It's possible to generate large data sets very quickly with metabarcoding. I went in with the expectation that it was going to be expensive and time consuming and it was going to have to be way better than microscopy to justify its use. What I came to realize is there's very little difference in the investment of time and money between one sample and 1,000 samples and somewhere over here 10,000 samples with the metabarcoding method. With microscopy, every additional sample is an additional unit of investment on top of it. And Sam talked earlier about optimization time with metabarcoding, and he's right, but I just want to point out that once you've optimized once for your system, you have a protocol. You don't need to optimize again. It's repeatable. And as we've shown here, it's better in terms of detecting interactions per sample that goes in than pollen transport by the traditional method. We know that's better than flower visitation, so to my view that makes it the best method so far if you want to generate lots of interactions from your number of samples. This study obviously focused on moths, which are one understudied pollinator group. There are others out there. I've put a wasp on ivy here. Andrew showed us with hoverflies yesterday. And it's also better at narrowing things down to taxonomic species, which could be good in systems where you don't really know very much about what plants might be involved in these interactions as well. Finally, there's this growing area of research which I find really fascinating that suggests that coevolution can help to structure networks. And even there's a very recent paper that's gone up on bioRxiv suggesting that nestedness in networks might be nothing more than a spandrel of coevolution, might be generated by coevolution between interacting species. To investigate that further, you need really two sets of data. You need interaction data and you need a phylogenetic tree. If you go at your interaction data through a metabarcoding method, you're generating the sequence data that you need to construct those trees. So you're killing two birds with one stone. I find that potential really exciting. So hopefully I've managed to intrigue some of you and get some of you thinking about possibly applying these methods. If you want to know more, if you want to talk to me about it, I'm going to be around for the rest of the conference, or contact me, email, Twitter, however. Thanks very much.
I was just wondering, so for the species that you didn't detect, did you check that they could be amplified by the primers that you used for RBCL in the first place?
Yeah. So one of the first things we did when we used a custom pair of primers. So we wanted to check that really carefully, and we checked a really wide range of plants that we gathered off the University of Hull campus, and with a couple of exceptions that were very readily explainable. It worked for everything. And actually, the three that I put up there, two of them we did detect, but we detected in such low read depths that they got cut out when we removed everything that was below a certain read depth threshold in order to try and reduce the risk of cross-sample contamination, and they got cut out by that, but they were actually detected, just in very low levels.
And so what was the read depth threshold?
We used 50 reads, which was based on levels of things appearing in our negatives and things from our positive controls appearing in other things. 50 reads would seem to be the right level to set.
Very good talk. It's nice to see somebody having a lot of success with their methods. I definitely agree with what you were saying about the kind of each additional sample is a sort of additional unit cost. I was just wondering whether you encountered any sort of significant problems with the optimization process, or whether or not it was ran quite smoothly because you were looking at sort of pollen in a more, I guess, cleaner environment.
Yeah, we did have some problems. For a while, we thought we might have to abandon the project because we couldn't seem to get amplification of pollen from moth processes at all. That turned out to be a result partly of inhibition from moth tissue being in there, so we had to wash the pollen off and then take the moth tissue back out before we did anything with it. And partly a function of the fact that pollen was there in very low quantities, so we had to really go way off the chart in terms of the amount and the concentration of the DNA extracts that we were using in our PCRs. So yeah, we did have quite a lot of. I think I was working on this for about 6 months, and the last 2 weeks of that was going from raw samples to sequence data.
Yeah, that's basically the sort of
thing
I found as well.
Okay, sorry, we don't have any more time for questions. We'll just move on to the last one. Thank you very much. Okay, so our last topic today is by Dirk Sanders, talking about artificial nighttime light changes and aphid parasitoid dynamics.
All right. Thanks for staying until the very end this morning. I would like to report on a field experiment where we manipulated the intensity of different artificial lights at night on a aphid parasitoid network. So we all know that light pollution is a very widespread phenomenon. It's emittance of artificial light at night by human sources into the environment. And it's potentially affecting a wide range of species, because circadian rhythms are very important for a lot of species to determine the growth rate, the metabolism, how they overwinter, and so on. There's a lot of wide range of different intensities of artificial light that might be exposed. So, for example, measured in lux, we have 100,000 lux would be a sunny day in the night without any artificial light. We have very low intensity of lux. Normally, sort of under street light, we have around 20 lux to 30. And urban sky glow, so the glow you can see when we have a clouded night, is around sort of 0.1 lux. We know so far that a lot of species respond to artificial light, so they either are attracted or they run away. What we don't know really is a community response. So how do ecological communities respond to artificial light at night? Is it a whole community response? Do we have different, maybe just the trophic level responding in a certain way? Certain species, like we know from climate change, so far that some species respond in some way, other species in another way. Can it be a... Seems to be stuck again somehow. Anything I can do? Yeah. So it will affect, for example, the plant level. We can expect bottom-up effects. If it affects more the higher trophic level, we can affect top-down effects, competition between species. A really good model system to understand dynamics between species is the aphid parasite system. It's been used very often. We have a lot of knowledge about it. It's got short generation times, and the very nice thing about it is that we can actually follow every single direction, because between a parasitoid and an aphid, because it relates to, in the end, the parasite will lay an egg inside an aphid, develops, and we end up with the aphid mummy. So the number of mummies are the successful interaction between a parasite and an aphid. And you can train undergraduates to go out and count these mummies. Problem again? So these species are kind of building blocks, and then you can assemble a community. These are our experimental communities. We have 2 plant species, bean plants, 3 aphids feeding on the beans, and the parasitoids, specialist parasitoids attacking them, then barley, grain aphid, and the generalist parasitoid, so linking these 2 Foodweb modules. And we're interested in parameters like might be affected, like parasite efficiency, plant biomass, and at the aphid level, competition and growth rate in these species. I've used this species, the system in the past, and we've shown it is very stable initially, which is very nice for an experimental system. Normally if you put parasitoids and aphids together, they will just crash very quickly. But if you remove one species from the system, you get a so-called horizontal extinction cascade, because this increases competition at the herbivore level and will sort of feed in reduced efficiency of these parasitoids will finally go extinct. So you end up with a very simple system. So we're interested now how these communities might respond to different levels of artificial light at night, and what would be the main drivers for the change we see. In 2005, we've done an experiment just looking at street light intensities 20 lux, and we found that actually aphids and the parasitoids responding. These are the treatments, the gray lines are the treatments, light treatment and night. And we see that with negative impact of light treatment. Until it was sort of, that experiment was done in autumn, so September, November, October. In October we found that actually normally aphids goes, many aphid population goes sexual because the eggs are able to survive in harsh winter conditions. But under actually artificial light treatment, we found that actually they still reproduce asexually even in autumn. So another field experiment where we had 8 different treatments, control, and different levels of light intensity at night. All these cages that we built, they were arranged in 6 blocks on the field side, and we had a polytunnel to maintain all these insect communities, the plants, fresh plants every week. Now to the results. We look, for example, at one aphid species, the dominant aphid. You can see sort of typical population dynamics. They grow, they reach carrying capacity, and then populations crash. If you look at low-level lighting, here 0.1 lux, so like sky up to 10 lux, we can see that they're actually similar growth rate, but they don't reach the same level of carrying capacity. Interestingly, at higher levels of lighting, we see there's almost no difference to the control. So high level lighting seems to have a similar effect on the aphid population than just the control without any light. There's one outlier with the 50 lux that sort of does something differently, or compared to the low levels of lighting. Looking at growth rate, where we just look at sort of week 1 to 4, where parasitoids have no large impact on aphid population. We don't see any effect or any difference in growth rates in the different aphid species. But as I showed, carrying capacity for all species together, we sum them all up. We see again we have the control at low level lighting we have a negative effect, and high level lighting apart from the 50 lux treatment we have sort of no effect compared to control. So if you look at carrying capacity, you would expect that sort of that might be a bottom-up effect. So the first thing is to look at plant biomass, and we have the bean plants. Resource for these aphid species, and we see that they actually follow but it's ecology, it's messy. Field experiment, its problem is always a bit messy, so it's only a trend I can show you here. So there is apparently more plant biomass under increased light treatments. And so there's a difference between low lighting level and high lighting level. The idea is that they actually can do sort of keep on doing photosynthesis even in the night and increase plant biomass. You can see some response in these aphid species here. They have lower densities at low level lighting and higher density at high level lighting. And the other plant species, barley, shows a different pattern. But again, we have sort of a positive effect of lighting on the plant level. And the aphid is completely responding in an unpredictable way somehow. So it seems that we actually have, lighting has an effect on the plant level. At higher intensities, we get more plant growth. At lower levels, they seem to suffer in some way. Now let's look at the parasitoid level. We see that parasitoid actually control these aphid populations. So if you see this is parasitism rate, it's the control, and starting week 4, and in the end of the experiment actually have killed most of the aphids in these communities. And we see a trend, at low level lighting we have a trend of a higher parasitism rate, at high level lighting is completely the same as control, without any light. If you average across, take the time out, just average, you see that low level lighting has a higher parasitism rate than high level lighting. And we understood how these parasites actually respond to artificial lighting. And we set up experiments where we looked at the functional response. So you give parasitoids different host densities, and time to attack these, and see how much they can actually extract the resource. And again, we found that lighting, so you have the type 2 function response. Black is the control without any lighting. We see that actually light at night seems to increase the attack rate. So it almost doubles the amount of hosts they can attack. In the literature, it seems to be that parasitoids are mostly active dawn and dusk and during the day. So if you have light levels increase their range, they can actually attack the hosts. So to conclude, we've seen that there's some evidence that artificial light at higher densities is actually enough to increase plant biomass, and we get bottom-up effect. Of no evidence for any growth effect on growth rate of aphids, so they all respond the same way. I don't know what's going on with computation. It has to be tested. And we have increased parasitoid efficiency on a certain. This might actually be true. The problem is with the whole community approach is that it's hard to disentangle the effects. So you have a bottom-up effect and top-down effect going on, might end up with no effect at the aphid level in the end. So to summarize, we've seen that artificial light indeed does affect the system, and that light intensity is quite important. The problem is, in this experiment, we had a lot of treatments and not enough replication. So we need to explore this more and the mechanisms. And the next step would be to do individual species responses and compare this in a model context to what we see in the whole community context, and compare what actually do individual species, what are they doing when they are embedded in a more complex community. Thank you very much.
Hey, do we have any good questions? Everyone's after lunch? Okay. Well, thank you very much to all the speakers of this session. I think it was very interesting, and yeah, definitely learned a lot, and I'm glad to.
Sorry, was the battery? Oh, okay, right.
Thank you.
Thank you.
Man, yeah, it's slow. You don't need it.